Okay, for the rest of this chapter, we're going to look at how the cell cycle is controlled by chemicals. So all of this like movement from prophase to metaphase to anaphase and so on and so forth, and from interphase into mitosis, all of that is regulated by chemical signals. So this also relates in cell communication. And so the frequency of cell division, how often cells divide, varies depending upon the type of cell. For instance, muscle and nerve cells do not, like nerve cells or neurons that we just learned about, do not really divide, whereas your skin cells divide all the time. So, so depending upon that type of cell, it varies with the chemicals that are produced in that particular cell. So these cell cycle differences result from the regulation at the molecular level. So we're talking about what kinds of molecules are made. So really it's governed by your DNA, which certain genes are turned on in some cells and not in other cells. The cell cycle appears to be driven by specific chemical signals. That we'll learn about these specific chemical signals present in that cytoplasm. And so we're going to look at some experiments that prove or give support for this hypothesis. Some evidence for this hypothesis comes from experiments in which cultured mammalian cells at different phases of the cell cycle were fused to form a single cell with two nuclei. So if we look here, what that means, there's a picture that illustrates that. Get to the right picture here. This is the picture that illustrates that. So you should be looking at this. Experiment number 1. If we look at experiment number 1, what they did is they took two cells and fused them together. These two cells were in two different stages of the cell cycle. So we just learned in the last video about interphase being made out of the G1, S, and G2 phases. And that mitosis, the M phase, is the prophase, prometaphase, and so on and so forth. So what we have here are two cells. This cell right here, I'm going to outline in blue, is the S, a cell in the S phase of interphase, and this cell in green is in the G1 phase. So they're in two separate parts of interphase. They're both in interphase, just different parts of interphase. And so then notice here when they fuse them together so that it's one big cell with two nuclei, notice that this nucleus still behaved as if it was in the S phase, but notice this cell now goes into the S phase as well. All right, so that could be, so when they're thinking about that, so S follows G1, so is this one just progressing to the next phase from the G1 to the S phase to the G2? But this one didn't progress to the next phase, so why is that? So then they did another experiment where they took one in the M phase. I'll circle this. In the M phase, and then this one, second one, is still in the G2 phase. And notice what they did is they took, G1 phase. They took one that's in further in the cell cycle. So it goes, remember, interphase, which was G1, S, and G2 phase. Remember that order from this picture? Interphase, the G1, S, and G2 phase, and then the M phase, mitosis, that consists of prophase, prometaphase, metaphase, anaphase, telophase, and so on. And so that's your M phase. And so notice what happened here is that when they fused them together, the one that was in the G1 phase immediately went into the M phase. It seemed to skip the S and the G2 phase. So this one skipped S and G2 and went immediately into M. Whoops, wrong color. M. And so why is that? Does anybody have any ideas as to why that is? Thinking about what we talked about just a few minutes ago, that the cell cycle appears to be driven by specific chemical signals, that the M phase, there were certain chemical signals in the cell that triggered it to be in the M phase, so that when they fused together, those chemical signals diffused, and this cell then behaved as if it was in the M phase because it had those particular chemicals. And so the same thing happened over here. The S phase had certain chemicals produced, and when they were fused together, those chemicals can diffuse to the S phase. And so that was the first indication or support that the cell cycle and what phase you're in are controlled by chemical signals within the cytoplasm of the dividing cell. So let's look at what these chemical signals may be. So the cell cycle control system. The sequential events of the cell cycle are directed by a distinct cell cycle control system, which is similar to a clock. The clock has specific checkpoints where the cell cycle stops until a go-ahead signal is received. So let's look at this on a picture. All right, so here's the cell cycle. So it starts here at the G1 phase. That's the first phase of interphase, then the S, and then the G2, and then the M phase, which is mitosis. So here we can see that there are 1, 2, 3 checkpoints. So let's look at here, the G1 checkpoint. This is often called the restriction point. This seems to be the most important checkpoint. Basically, they found that if a cell gets past this checkpoint, most of the time then it will continue and actually divide and get past the other 2 checkpoints. So I'm going to put here restriction point, a go here usually completes the cycle. And what the checkpoints are doing basically is checking to make sure that up until that point things have happened properly. So in the G1 phase, the cell is beginning to grow. So G1 checkpoint is the cell behaving properly? Is the cell growing? Because the cell grows before it divides. So it's basically checking to see, is it doing what it's supposed to do? If it seems to be behaving properly, it gets past that checkpoint and goes goes to the next phases, which the S phase, which remember is copies the DNA, and then the G1 phase. So the second one is this G2 checkpoint. And like one of the things that it checks for is, did the DNA copy correctly? And did it copy at all? If it didn't, then something's wrong with the cell. We don't want it to divide, so we're going to stop and not let it divide. And then the M checkpoint, the third one, they check to see, did the chromosomes line up correctly? C-zomes. I'm going to abbreviate chromosomes, C-zomes. Did the chromosomes line up correctly? So for many cells, the G1 checkpoint seems to be the most important one, as we just said here. So if it gets through all of these, the G1, it usually gets to the G1 and or G2 in the M checkpoint, and then it will actually divide. So you start out with one cell and then you'll end up with two. And if it gets that go-ahead signal at the G1 checkpoint, it will usually complete the S, G2, and M phases and divide. So it usually, that's what I meant by it usually completes the cycle. But if it doesn't receive the go-ahead signal, it's going to exit the cycle. It's going to not stay in that cell cycle, and it switches to a non-dividing state called the G0 phase. So the G0, so it can, if it doesn't get past this, it can enter the G0 phase. So let's look at this, what this little picture here is showing you. So this part right here is that G1 checkpoint. So they're only showing you this part of the cell cycle. So if it receives the go-ahead signal at the G1 checkpoint, the cell continues on in the cell cycle. So if everything's happening properly, it's like a doorway. It'll let it go through, and this is a little stoplight, and it has the green light. If I'll color that green, it has the green light and can go. If it does not have the go-ahead signal, if something's going wrong with it and it didn't grow properly and it's not behaving properly, it gets the red light and enters into that G0 phase. That is the non-dividing phase. So actually, if we write a few things here, most human cells, most human body cells are in the G0 phase. If you think about, most of our cells are not continually growing throughout our lifetime. They grow for a certain amount of time and divide for a certain amount of time and stop. And so let's talk about mature nerve and muscle cells. I said this at the beginning. Stay in the G0 phase. So they're in there. Interesting, liver cells can be what kind of called back. I'm going to put in parentheses. From G0 and continue the cell cycle at certain times. And so what would be a time when that would be occurring? So during injury. So during normal time, mature liver cells are not dividing, but they can start to divide. And then there are some cells, like skin cells, skin cells always continue to divide. We are constantly sloughing off our skin cells. They're all in the air around us. So these guys are not in the G0 phase. So that gives you 3 different examples of some different types of cells in the body. So let's talk about one go-ahead signal that happens actually here at the G2 checkpoint and triggers the cell's entry into the M phase. So the cell cycle, we have cyclins and cyclin-dependent kinases. That word kinase should seem familiar. ACE, it's an enzyme, right? And kinases, remember what they do? They phosphorylate. Remember that these were enzymes, if you recall from chapter 11. that phosphorylate a molecule. So two types of regulatory proteins are involved in cell cycle control. We have cyclins and the cyclin-dependent kinases. We're going to call those from here on out CDKs. The activity of cyclins and CDKs fluctuates during the cell cycle. So we're going to look at one type of CDK called MPF. And so we have cyclin and MPF. I'm going to put here, this is a CDK. Put that in parentheses there. So that's what MPF is. So if you notice here, let's look at the relationship and where we're at. So they're showing you time here. This is cells repeatedly going through mitosis. So this is dividing, and then you end up with two cells. And then those two cells enter the G1S and G2, and then the M phase and divide. And then those cells enter the G1S, G2, and divide through the M phase, and so on and so forth. So this is time and what happens with subsequent cell divisions. So you can see here in the M phase, what happens is if we look first, the cyclin is going up. And then if you notice, MPF seems to go up afterwards, almost because of that increase in the cyclin. So it goes up and MPF goes up. Then notice here, cyclin goes down, drops down, and MPF drops down. And then notice here, the MPF drops down, and then its MPF isn't in the cell for a while, but the cyclin, here we go, cyclin drops down here, then the cyclin goes up. Notice it starts to go up to kind of the end of the S phase there and progressively goes up, and then the MPF activity follows. And then cyclin goes down, MPF goes down. And does this make sense? Let's think about this. MPF is a cyclin-dependent kinase. What that means is it needs cyclin to be active. It's dependent on cyclin. So let's write here down a few things down here. So MPF is a cyclin-dependent kinase. Let's talk about what that means. So it needs to be attached to a cyclin to be active. They call it MPF. It's the maturation promoting factor, but sometimes they think of it as the M phase promoting factor, so we can write maturation or M phase promoting factor. What it does, so let's write that, why they call it that. It triggers the cell's passage past the G2 checkpoint into the M phase. And that's why they call it MPF. So let's look at this in a different way on the back. So if you look here on the next page here, this cycle here. So here we have let's look at here the beginning of the cell cycle, G1 and the S and the G2. So G1 phase, this is the beginning of the cell cycle. So here we have CDK that's found in the cell, and the CDK is in the cell in the G1 phase, is in the S phase, but notice CDK, it needs to be attached to a cyclin. That's why they call it cyclin-dependent kinase. It needs to be attached to a cyclin in order to be active. So right now in this phase, CDK isn't active. It doesn't work, but it's hanging out in the cell. It's present in the cell. So it's present all the way through into the G2 phase. We still have the CDK. It's inactive. Why? Because there's not cyclin. But notice what happens towards the end of the S phase. We see here, what color did I use with cyclin over here? I use cyclin as red. So notice here, cyclin starts to, if we look here at the G1S, towards the end of the S phase, cyclin starts to accumulate in the cell cycle. That's what we see over here. The cyclin is accumulating. The cell is making cyclin, so that's my red here. And here's my red cyclin. And so now as it accumulates, and they remember with diffusion, they're bouncing around, moving around, they have kinetic energy, they come together and bind together, and now we have an active CDK. So if we look, let's make this one green. So here's your CDK, let's make him green. And the cyclin was red. So now you have your red cyclin bound with your green CDK, and now the CDK, it's a cyclin-dependent kinase, now it's active. So this is, so we can write here, let's write here at the arrow what happened here. So cyclin binds to CDK to form MPF, which is an active CDK. So now it's active. It has activated it. So this is now in the active form. Active CDK. And what this is going to do when it's active is this is going to trigger entry into the M phase. Triggers entry into mitosis. And so then notice it gets past that G2 checkpoint. So this G2 checkpoint is driven by whether or not MPF is present or not. So then notice that it'll go through the M phase, and then the cyclin, which was red, the cyclin is degraded. So now we have, so there are enzymes that do this in the cell. All right, so let's put here by enzymes. And you have left over the CDK. And the CDK stays in the cell and in the inactive form until cyclin accumulates again, forms MPF, triggers entry into the M phase, and the same thing happens over and over and over again. So this here, so the cyclin accumulated, and then when the cyclin accumulated, it bound to the CDK, forming MPF. That's why over on the back side, as the cyclin accumulated, eventually here's the MPF accumulates, then the MPF goes down because the cyclin is degraded by enzymes. Right here, this part of the graph is the cyclin degraded by enzymes, and then immediately after the cyclin is degraded, MPF is no longer there because it's not in the active form anymore because it's now the inactive CDK. And so, and it stays down until the next part of the cell cycle, the next cell cycle. And so all the different checkpoints are controlled by different CDKs. So we just talked about the G2 checkpoint, but the G1 checkpoint has, they think, roughly about 3 CDKs that help to regulate it. And so each of the checkpoints have their own kind of molecules. So this is one example of chemical regulation past a checkpoint. All right, so let's continue on. Stop and go signs, internal and external signals at the checkpoints. An example of an internal signal is that kinetochores not attached to the spindle microtubule send a molecular signal that delays anaphase. Let me talk a minute about what that means. If you have, remember, kinetochore microtubules are microtubules that attach to the chromosomes. So if you have, here's one side of the chromosome and here's another side of the chromosome. Remember, during how they line up is the spindle. These guys are called kinetochore microtubules because they actually attach to the chromosomes. We have also non-kinetochore microtubules that help to elongate the cell. And so, so these guys, these kinetochores, are supposed to be attached. So if they're not attached yet, then they send a molecular signal that says, hey, don't trigger anaphase yet. Remember, metaphase is when the chromosomes line up single filely down the center of the cell. And if you have, here's your chromosome, and they're not attached yet to that, they send out chemical signals that say, hey, we haven't attached to our chromosomes. So these are in green here, are the chemical signals that they're sending out, saying we haven't attached to the chromosome. Those chemical signals delay anaphase, which, remember, is when they pull the two copies, two sides of the chromosome to opposite sides of the cell. And so they do that, and so you don't want that to happen when it's not attached. So that's what that means. So that will delay and kind of stop the progression of the cell cycle. Some external signals are growth factors. So external signals. So this was an internal signal going on inside the cell. That's right, growth factors. Some are molecules that are released by other cells. So some external signals are growth factors, which are proteins released by certain cells that stimulate other cells to divide. So let's give a specific example. So, for example, platelet-derived growth factor, or PDGF, stimulates the division of human fibroblast cells in culture. So this has been found to be a factor that's released by one cell that communicates with another cell to trigger it to divide. So let's look at this as an example in this figure on the next page here. So what they're showing you here is a petri dish, and they're taking a sample of connective tissue they're cutting it up with scalpels. So we can write here that they cutting connective tissue into small pieces. That's what that's illustrating. Then they mixed those small pieces, enzymes digest, the extracellular matrix. which remember is what if you have two cells, it's kind of like fibers and strands that connect the cells, and they're bound to the cell membrane. So it digests all of that, so you have free-floating cells that aren't connected to each other. So, which means that you get a free-floating cells from that. So that's the purpose of this. And then they took the cells and they grew them on petri dish plates. And what they're growing them on is the growth medium. The growth medium has everything that a cell needs to survive. So it has some food and water and different things that it needs to survive. So it's kind of like a gel there. And there are cells on it. And so it's hard to kind of see here. Cells on it, and these two are the before. So let's put these in different colors here. So this is before, and this is before. So to begin with, they plated the same type of cells on two different types of plates. Two different types of plates, one with that growth factor and one without. So they actually put the growth factor as part of that growth medium that they were growing the cells on. So it had food and so on, and this one had that growth factor, the one that we talked about over here on the other side. Oops, I guess it wasn't on the other side. It was here. All right. The PDGF, the platelet-derived growth factor, stimulates the division of human fibroblast cells in culture. So these cells are human fibroblast cells, and so that's the before. They look the same before, and then they incubated them and let them divide, and what happened is without the PDG, it pretty much looks the same as it did before. But with the PDGF, I'm going to put a bunch of them in here, there's lots of growth. So this is the after, so I'm going to put here, this is the after without PDGF. So we can see here that these two, color coding helps so that we can see before and after what happened with these two. And then this is the after with the PDGF. So before and after. Obviously, huge amounts of growth there. And so what is the conclusion? The conclusion would be what? What would you say? I would say that the PDGF stimulates the division of, remember these were the human fibroblast cells. And this is the actual real picture of that. So that is an external signal that can trigger cell division. So there's internal signals, there's external signals, there's internal signals that stop things from the cell from dividing. And so there's lots of different signals that are all playing a role in controlling cell division. All right, so let's continue on. What other things play a role? Another example of external signals is what we call density-dependent inhibition, in which crowded cells stop dividing. Most animal cells also exhibit anchorage dependence, in which they must be attached to a substratum, which is a surface. They must be attached to something in order to divide. So they stop dividing if they get too crowded and there's not enough room, and they stop dividing if they're not attached to anything. So let's look at this back picture here on the other side of the page as far as what is going on with these cells. And so what this picture is trying to show you here is that in this picture, cells are anchored to the bottom of this dish. So they're growing cells in a little dish again, just like the previous picture. So they're attached to something and they're dividing, and that is anchorage dependence. And so then they will divide, but then they stop dividing, they make a single layer. So that's what this is supposed to be showing you here. They have all these different cells forming, let's just draw them in there, it's hard to see. All these different cells that form a single layer along the bottom of the petri dish. And then they stop dividing. So when they stop dividing, they're exhibiting density-dependent inhibition. So this is how normal cells behave, where they divide when they're attached to something, and then they keep dividing until there's no more room, and they stop dividing. So then they enter into that kind of G0 phase. And then notice here what they're doing here is they're taking a little spatula and scooping out some of those cells, and so there's a little hole here. So they're scooping out a couple cells, so you have all the cells in here. But we have a little hole here, right here, where they scooped it out. And notice what happens is the cells will then trigger, the cells around it will trigger them to divide, and they fill in that hole and will stop dividing once they get crowded again, and so therefore exhibit again density dependent inhibition. So that is how normal mammalian cells behave. So let's look at this next picture here. The next picture is about what abnormal cells do. So this picture is of what cancer cells do. So cancer cells do not exhibit anchorage dependence or density dependent inhibition. There seems to be nothing that stops them. They don't have to be attached to a surface, and they don't stop dividing when they get crowded. And so you can see here that it just takes one abnormal cell, and that one abnormal cell divides and divides and divides, and not exhibiting either of these mechanisms that are in place in normal cells, and you get a mass of cells. And when this happens inside the body in a particular space, we call that mass of cells a tumor. And so basically, cancer cells lose control. There's a loss of the control of the cell cycle. So that's what this next little bit is about, is the loss of cell cycle controls in cancer cells. Cancer cells do not respond normally to the body's control mechanisms. Cancer cells form tumors, which are masses of abnormal cells within otherwise normal tissue. If abnormal cells remain at the original site, the lump is called a benign tumor. So they stay where it originally was. If we look at the next page, malignant tumors invade surrounding tissue and can what we call metastasize. And what that means is it takes the cancer cells, and usually they get into the bloodstream and can be taken to other parts of the body where they can form what we call secondary tumors. So what this picture is showing a picture of breast cancer and how this works in breast cancer. And so in the breast tissue, a tumor is formed. And remember that you only need, because what a tumor is doing is growing abnormally and basically bypassing all those checkpoints. So the G1, the S, or the G1, the G2, and the M checkpoint, it doesn't stop. Remember back when we looked at this, it's supposed to stop here and enter the G0 phase so that if the cell is abnormal, it doesn't ever get past the G1 checkpoint and it doesn't even get past any of the checkpoints and doesn't divide. But cancer cells bypass those checkpoints and are able to divide anyway, even though they're abnormal. And then when they start to divide, they don't exhibit anchorage dependence or density dependent inhibition, and they kind of divide uncontrollably. And so then what happens is you get this tumor. So there's the tumor. And then if left unchecked, those cancer cells can continue to divide, and they start to, as it says here, invade the neighboring tissue. So it starts to be invasive into the tissue. If left unchecked even more, they start to spread, and they can actually enter, because cancer cells don't have to be attached to anything, so they can break away from the tumor. So sometimes that's what they do, and they can get into your either lymphatic system, remember your lymphatic system with your lymph nodes and so on, or they can get into your bloodstream. Either one of these vessel systems go throughout your whole all of your body, and so that those abnormal cancer cells, that's what this is supposed to be showing you here. If the tumor is in red, if I'm going to do the tumor in red, this is the cells in the bloodstream and in the lymphatic system. They can get in there. And those abnormal cells can go and be taken throughout your whole body, and then randomly they can leave the blood vessel or leave the lymphatic tissue at another part of your body and attach. So it leaves the bloodstream and attaches somewhere else. It can attach to an organ or something like that. So something that can start out as one type of cancer, let's say on your kidney, can spread and can get into your bloodstream and attach to your lung, and so then you have lung cancer. That would be a secondary tumor. That's what we mean here when they form secondary tumors, and so on. So, and often when that happens, we say that the cancer has spread. That's what in layman's terms what they say, or we could say that the cancer has metastasized, where it exports cancer cells through via the bloodstream or the lymphatic system to other parts of the body. And this is why it's really important to be able to find it early. When they say detect cancer early, what we're talking about is detecting in one of these stages before it actually gets to the point where it can metastasize and get into your blood vessels and so on. You want to catch that beforehand. And so cancer cells are examples of cells that don't exhibit all the things that a normal cell has in place to keep that cell cycle in check. And cancer cells kind of escape that. Okay? So we're going to write a few other things on the bottom of this, some things about this. So transformation. Right here. This is the process that converts a normal cell to a cancer cell. So let's talk about what can cause this. Let's let's write here, mutations can cause this. Mutations in certain genes. And let's write mutations. Let's write what this is. This is a change in your DNA. So the order of the bases, the A's, T's, C's, and G's in your DNA are the basis for that. So mutations in certain genes due to, what causes a mutation? Due to chemical carcinogens, carcinogen is any chemical that is known to be cancer causing. X-rays can cause this, and other high energy radiation. And lastly, and some viruses are known to cause cancer. Like, so cervical cancer has been linked to viruses. All right, and I want to talk about two different types of genes, and then we'll be done here. So there's a type of gene called a Proto-Oncogene. These guys code for proteins that stimulate normal cell growth and division. So it stimulates normal cell growth and division. So can you see that if this type of gene was mutated, that it wouldn't stimulate normal cell growth and division, and actually can lead to cancer if one of these genes are mutated. So mutations to it can cause them to become an oncogene. This is a cancer-causing gene. So the proto-oncogene stimulates normal growth, cell growth and division. Remember, cancer cells, so what they do is they kind of basically divide uncontrollably. So the mutation that actually happens to these doesn't actually turn the proto-oncogene off. It just stimulates it and increases either the amount of the product that's made by that gene or in the activity of it. So it makes it actually stimulates it and does excessive cell growth and division. So that's the next thing I want to write here, is that the mutation can lead to an increase in either the amount of the proto-oncogene's product or in the activity of each protein molecule. All right, and then we have the tumor suppressor genes. these guys, as they might sound here, suppress or code for proteins that inhibit cell division. So this promotes cell division, this inhibits it. So they code for proteins that inhibit cell division. So let's write how they do this. So they prevent uncontrolled cell division. So let's talk about how this happens, what happens with cancer cells. You can see that if this gene, one of these genes, is mutated, then that's going to decrease the activity of this gene, which may cause the cell to divide. It's going to grow and divide because it's not being suppressed. Because this suppresses, it stops cell division. So if this is mutated, then the cell isn't stopped and therefore can divide anyway when it probably shouldn't. So let's write this. Any mutation that decreases the normal activity of a tumor suppressor gene, or sorry, protein, may contribute to the onset of cancer. And how that basically works is, I'm going to put in parentheses that it's stimulating, that mutation would be stimulating growth of the cell through the absence of suppression. So it's not suppressed, and so it can divide. The absence of suppression. And that is mitosis in Chapter 12.